Yes, hello, thank you. So first off, I would like to thank Max and Ivan for having me here. I'm extremely excited to be here. This is, I think, just a fantastic opportunity to discuss across disciplines and to talk about some really pressing issues in biology. I'm going to change gears a little bit. So a lot of what we've seen so far this morning has been about information being encoded in sequences, though not all of it. I'm going to talk about information in cell signaling. So the basic idea is that regardless of the kind of cell you're going to talk about, oh, and I should say that this is very short, so I'll do my best and not to go over. And also, it's a perspective talk, so I'm going to give you maybe a little bit more perspective. And I'd say the number one perspective I have to bring is that when thinking about information theory in biological systems, it's often helpful to think about the physiology of the system that you're talking about. Like, what does it evolve to actually do? Because that might allow you to cast the questions in a way that's perhaps relevant to the design principles or the observed structures that you see. So anyway, the point is that almost every cell you could think of lives in some kind of environment, right? So for unicellular organisms, they're in the environment. And there's information in that environment that they need to know about. So for instance, if I'm a yeast cell and a new nutrient becomes available, I might want to know about that so that I can express the enzymes that would allow me to import that nutrient and then eat it. In a context of multicellular organisms like us, the information that's in the environment is largely about the state of the tissue. It's largely generated and maintained by other cells. So it's actually encoded in the concentration of cell signaling factors, largely, though not always, things like hormones and cytokines and stuff like that. And the point is that that information is actually how cells communicate with one another. And that cell-cell communication is how cells sort of self-organize their behavior within our tissues in order to achieve whatever sort of physiological goal they have. And I'll talk about one example of that. But the basic idea is that there's a problem, right? Because the information's on the outside of the cell. And there's this barrier that has to be there. That's what defines cellular life, a cell membrane, cell wall, regardless of the system you're talking about. And the way that this problem is overcome sort of almost universally is that there's an integral membrane protein called a receptor that lives in the cell surface that interacts in some way with that information. Usually the information is encoded in the concentration of some ligand, so basically the concentration of some molecule. That molecule binds to the receptor and induces a conformational change that can be read out on the inside. And often downstream of that event, there's a complicated protein interaction network that somehow transmits and processes that information. Ultimately leading to the activation of other proteins called transcription factors, which control gene expression, and also large-scale changes in cell fate and cell behavior. So for instance, one thing that can happen is you treat a cell with a certain signal, and it will spontaneously undergo programmed cell death, apoptosis. Other times it might differentiate. There's lots of things that can happen. There's an obvious, and I think very natural, sort of application of information theoretic frameworks to this problem, both at the level of the cell-cell communication that's up here and also at the level of this particular channel, which is transmitting this information from the outside of the cell to what has to happen inside. And Andrei Levchenko's group actually, I think, had a seminal piece of work about 9 years ago now, where they actually, I think, kind of pioneered the application of information theoretic thinking to this kind of system in biology based on a particular kind of signal called TNF, which is involved in inflammation and other processes inside of our body. Essentially what they could do is they could treat cells with these particular values of the signal, so changing the concentration, changing this input concentration here, and then measure for each individual cell how much of a transcription factor called NF-kappa B got activated after a certain appropriate period of time. And what you can see here are the results. I don't have time to go into this in much detail. And anyway, Andrei could tell you about it. The different levels of TNF are here. And you can see these different distributions of responses. So this is naturally an input-output relationship. And we can apply approaches developed in information theory to kind of quantify the amount of information that's transmitted between these different levels of TNF and these responses. And one thing you can note here is that there's an overlap. So if we look at this distribution of transcription factor activation in cells treated with this sort of saturating dose of TNF, you can see it overlaps actually with the distribution that we observe in cells that are not treated at all. So as a result, when you actually plug this data into an estimation algorithm that tells you something like a channel capacity, you get less than 1 bit. which is surprising because we expect that these cellular systems have evolved to gain information about the environment. And it turns out that there's a lot of heterogeneity between isogenic cells in these kinds of populations, and that seems to limit their ability to actually do that. And it's not just TNF signaling to NF-kappa B. The Levchenko group and others have looked at this in a variety of different contexts. And you see actually that in many different scenarios, with many different signaling molecules and many different responses, you get less than 1 bit. And I think this has led to a really productive conversation in this field about, how do cells ever do anything reliably, right? So if they have so little information about their environment, how do they actually make reliable decisions? So they may be integrating information that's been shown, well, not shown, but I think suggested by a couple of different labs. Maybe they're integrating over time. Maybe there are multiple pathways that are kind of collecting information and comparing. Maybe systems compensate for noise. But one of the ideas that we had was to actually look at this in a physiological context and think about, what kind of processes these types of signals are controlling. And one example would be that situation where a signal controls cell fate decisions. So this is I'm not going to go into the details. This is a signaling network inside of cells. This is our extracellular signal. It's related to TNF, but it causes cells to die. So it's the TNF-related apoptosis-inducing ligand. It binds to the cell surface receptor here, and then there's a sort of a simplified schematic of the protein interaction network downstream. And the cool thing is that we know a lot about these interactions, and we can characterize the activity of, in this case, not transcription factors, but cell signaling molecules that are involved in this process. Specifically, this initiator caspase, which is like the first thing that gets turned on. Not exactly, but it's upstream. And then the effector caspase, which is sort of the cell executioner. And we can look at these activities in individual cells. There's obviously here a clear phenotypic response, right? So we can tell if a cell is dead or alive. In an assay, you can easily tell, oh, well, this cell is dead and this cell is still alive, so we can know something about that. And the other thing that's interesting, and it's something that because of time I've sort of glossed over, one of the very intriguing factors in looking at cell signaling from an information theoretic perspective is that when people go in and tag these molecules with a fluorescent marker or do some measurement of, say, the concentration of these different proteins in different cells in a population, you often see very wide distributions, often log normal distributions. So there's a lot of heterogeneity there, which is one of the reasons that we see such low information capture in these systems. And that's true here. So several of these proteins have been studied this way, and you see the same thing. So what we did was a very similar experiment. I should say, this was all done by our collaborators, Peter Sorger's lab at Harvard Med School, specifically John Bachman in his lab. They did this experiment, and so what they did is they have different values of trail. This is hard to see. It's not the best situation here, but basically what we saw is precisely what the Levchenko group saw with NF-kappa B. So if we look at the activation of these proteins as a function of different concentrations, you see there's significant overlap between the lowest 0 concentration and a saturating dose, and we get a little bit less than 1 bit of channel capacity. If we look at this executioner molecule, another sort of downstream point in this pathway, we see that there's even less information. But when we flip this on its head and say, well, actually, what this molecule is trying to control is not necessarily the response of an individual cell, but rather the percentage of cells that die in the population, get a very different result. So what we're doing here is it's the same exact experiment. We're just looking at a different version of it. So we add different values of this ligand, and we look at the cells in the population that survive. And what we see here is that as we add more and more ligand, this level of surviving cells decreases up to a particular point. And this type of experiment is far less heterogeneous and far more repeatable. So rather than seeing these really wide distributions where you get the same, you get overlap between maximal and 0 dose, here when there's no trail, none of the cells die, and you can see actually you get nice separation between these things. So when you plug this into the same kind of estimating algorithm, you get much more information. And The reason for that is actually extremely simple. It's a phenomenon that's known in physics as suprathreshold stochastic resonance. The basic idea here is that we can sort of look at that in a simple model where each individual cell has a response that's a deterministic function of the signal plus some noise. And the point of this model is not to model what's actually happening inside the cells, but just to explicate the basic phenomenon, the very simple phenomenon that's going on, or that we think is going on here. So the basic idea is that in this kind of model, every individual cell has a deterministic piece which sets the average response, and then there's a noise term that's simulated independently for every cell, and we might draw that from a Gaussian distribution, say, with some sort of well-defined standard deviation. And so that generates some error bars here, and you can see this is a very noisy response. If this is the molecular response of the cell and this is the amount of the signal that is added, you can see there's quite a lot of overlap there. There's not going to be a lot of information. But if we also define a threshold between alive and dead, has to be a threshold, right, in order for this to be both, either alive or dead, we see that actually the percentage of cells that are above that threshold varies nice and smoothly. So if we plot this in a different way, the fraction of cells that are alive, when there's absolutely no noise, this is a step function. As we add noise, we make this transition more gradual. And the error bars here, which define the sort of, if you did this multiple times, how similar would the number of cells surviving in any given signal be, that's set not by the noise properties of this system, but actually just by the binomial, how many cells you have. And so the more cells you have, the more information you can encode. And that's just shown here. So there's this natural trade-off between the amount of information individual cells have and the amount of information that's available to the populations. I'll skip through this. One interesting question that we had that sort of arose from thinking about this kind of problem is how might this noise be regulated? So I don't have time to go into it, but we saw that in certain contexts there's actually quite a lot more information available to cells. So it seems like there might be something happening where in certain cases you need to have noise in order to control populations, or at least that's a hypothesis, but in other cases you might not want that. So one thing that we're very interested in, which I think Werner's talk sort of suggested, is thinking about the gene regulatory networks that actually generate the heterogeneity that we observe. So as I said, the levels of proteins that are involved in cell signaling vary between, I have to go quickly, they vary a lot between cells. And we think that there might be something going on in terms of how the gene regulatory networks actually regulate that. And you can see here that people are using new sequencing technologies to kind of characterize single-cell expression levels of proteins in an unprecedented way. So we're thinking about that. The other grand challenge, I would say, or the other, not grand challenge, I shouldn't be so grand, but the other challenge that's out there, I think, is thinking about how cells might compute and communicate through trajectories. So this has become quite common now for people to recognize that while we might look at one individual time point, if we actually measure different cells at different concentrations of these input, and instead of just doing it at one time point, look at the trajectory, we see some evidence that actually cells are distinguishing different signals or different levels of signals through features of these trajectories. But this is a complicated problem, actually, theoretically, because unlike we might have in certain engineered systems, it's unclear. It's not necessarily clear there's like a frequency domain that we have to think about or something like that. So there's a lot of work, I think, to be done here. We're thinking about it ourselves, but quantifying information in trajectories, I personally find it to be somewhat non-trivial in this context. And I'd love to talk to people here about that. Why I think it has to do with the Lebesgue measure and the fact that there isn't a Lebesgue measure or a translation invariant measure in infinite dimensional spaces. So I'm sure that that's been discussed a lot and thought about a lot in communications theory, so I'd love to learn about it. And with that, I'll just thank the people involved. Ryan Suderman, as I didn't mention, he was the graduate student in my lab at KU before we moved to UCLA. He did this, the computational work. And then, as I mentioned, John Bachman and Peter Sorger's lab. Thank you.
It's the record for keeping us on the schedule here. So we have time for one question.
Yeah, okay, cool.
Maybe right here.
Andre.
There you go. Thank you. Well, I want to say wonderful talk, but you mentioned my name so many times that I cannot do that.
Well, it was inspiring.
So I guess with the you said that the more cells you have, obviously the better, the higher amount of information. But basically, this is really, in your case, a choice between two outcomes. dead or alive, right? So in
Right.
a sense, it's kind of binomial. You
Right,
sort of
right,
mentioned
right.
that. So the error in a sense should scale as 1 over square root of n, right?
It
of
does,
the number
actually.
of cells.
Yeah,
So
yeah.
I just wanted to kind of, again, you will not necessarily, I mean, for a single cell, it would be sort of almost meaningless
Right.
and error prone.
Right.
And it would be interesting, again, to kind of revisit that scaling that you have with the number of cells.
Right, yeah, no, it is. It pops right out. It's obvious that you would get, so the amount of information it goes like 1/2 log base to n, right, where n is the number of cells for precisely the reason that you just mentioned. So it would be interesting to consider multinomial cases, but in the, you could, like, if a cell could decide between death or differentiation or quiescence or a variety of options. We haven't formally looked at it. I think you would get exactly what you would expect there. What you find a lot is that there's multidimensional signaling spaces that control that kind of information flow. So these guys don't live with just one signal, right? They're getting many, many inputs, and they have this multinomial set of outputs. We haven't looked at that much at all. It's a very interesting thing. I think one thing that I'm interested in is sort of figuring out, okay, what are the actual physiological like what's going on inside the tissue, so we can sort of inform that kind of question a little bit better. Should I stop? Yeah, I should stop. All right, thanks.